Hi, it's Mr. Polochak, and today we're going to be talking about a type of cell division known as mitosis. If you look up here, you can see that we've got in the 12 o'clock position a cell that's been given a signal to go ahead and divide. And this type of division, again, is called mitosis, and you can see as we move around clockwise, that cell prepares, it grows, it replicates the DNA, it grows a little bit more, and then it goes and it splits. So there are some characteristics about mitosis that you should be aware of, and one of them is that mitosis creates two new cells. These two new cells are called daughter cells, and they are identical to one another. It is a genetic copy, it is a clone, and the reason why that's important that we have that process is because this is going to be used in growth. This is how a zygote, which is a fertilized egg, is going to grow into a trillion-celled baby. We want to maintain that chromosome number so that one cell becomes 2, 2 becomes 4, 4 becomes 8, and so on and so on, and that's a part of embryonic development. The other thing that we see is tissue repair. Any type of injury that you may have sustained, this is how we heal. So mitosis helps with injury or tissue repair. This occurs in body cells or somatic cells. That word "some" means body, so if you look at a lysosome, that is a body that breaks things up with digestive enzymes. If you look at a chromosome, that is a colored body. And that's just how we use some of the parts of words to make sense of how they fit into biology. My goal for this video is so that you can identify the phases of mitosis and know some characteristics about each of the phases. So let's go ahead and talk about what happens before we go through the mitotic division. How does the cell prepare? And that's what we see here with the cell cycle. If you look up at the dial here, you can see, look toward the center. Most of the time a cell spends is going to be in interphase, and that scientists mistakenly called a resting phase, but the cell is not resting at all. You can see in that G1, that's a growth phase. It's preparing, it's getting larger, it might be increasing organelles. And then you get to that S phase, which is a synthesis phase. That's where your DNA is going to replicate. So we're going to make a copy of the DNA, then we're going to grow some more. Once you reach that point, that's a checkpoint, you can go ahead as a cell and start going through mitosis, which you see there in that reddish area, are made up of stages called prophase, metaphase, anaphase, and telophase. There are some cells, like nerve cells and some muscle cells, that don't divide anymore once they're formed and they're mature, and they enter what you see going across is that G0 phase, and they can stay that way. They're not going to enter interphase, they're going to stay as they are, and they won't divide anymore. So that's what the G0 means. Interphase, most of the cell's time is spent in that phase. The other mitotic phases happen pretty quickly. And what I'd like to do is go through some of those stages with you now. The first stage we look at, again, is interphase. It's not really a mitotic phase, it's really a preparation. But the reason why I bring this up is because if you look inside the nucleus there, you can see that if you look here, that the chromosomes are in this really thin form, okay? The chromosomes here are really thin. And this is where we're going to double that stuff. And it's in a form called chromatin. So I like to help my students remember that chromatin is thin. So if we had, a piece of DNA here like this that's thin, it's going to make a copy right here next to itself like that. So that DNA material is going to double by making a copy of itself. Once that happens, we reach prophase. Prophase is really the first mitotic phase. And the chromosomes are going to change. Instead of being in that thin form, they're going to shorten and thicken and form structures you may classically see, shaped like this. Okay, that's what we kind of see over here. Those are called chromatids, and they're copies. So this one right here, okay, this is the original, and the one right next to it here, that's the copy. That's the sister chromatid. Now these chromatids are held together in the middle in an area called the centromere. They're kind of shown as white spots, but this would be a centromere right here. Here's a centromere here. another one, and another one. So you can see we have 4 centromeres and 8 sister chromatids. That DNA has doubled. In prophase, the chromosomes shorten and fatten. They change from chromatin to chromatids, and the nuclear membrane, you can see, start to disappear. The other thing that you may have noticed is that we're getting these structures here called centrosomes, and they're starting to move apart and create these microtubules or spindle fibers. That's going to let the cell organize, splitting up of material. Okay, let's go to the next one. Now that we are at metaphase, we can see that our chromatids are lined up in the middle. We call this an equatorial plate right in the middle here, and we can see our centrosomes are at the poles, and we see these spindle fibers. Now, for AP biology, we have to know about these particular spindle fibers. They're called kinetochores, and they connect to those centromeres. Remember, the centromere is the spot right where the sister chromatids join. So here are the 4. And these kinetochores are really motor proteins that let the chromosomes walk away from one another as they move toward the poles. There are also, which are not shown in this diagram, non-kinetochore pieces. These are spindle fibers that will slide past one another. They do not connect to the chromosomes, as I'm drawing here, and they help push that cell apart so that we can orchestrate moving organelles and moving those chromosomes to opposite ends. So that's the difference between a kinetochore and a non-kinetochore microtubule. Now, scientists wondered, was it the centrosome that was, if we look over here, was it the centrosome that was kind of reeling in or pulling in those chromosomes so that they separate, kind of like a fisherman reeling in a fish? Or is it the chromosomes walking? What they did is conducted an experiment where they took a laser and they cut these spindle fibers or microtubules here, and they waited to see what would happen. If it was the centrosome that was reeling in these chromosomes, then by cutting them here, we should see them kind of stop. We should see a chromatid piece right here just stop, and this kind of wind in and nothing happens. But that is not what they discovered. Cutting these microtubules here, they did see that this chromosome did keep clicking and moving along until it reached the broken end. So that's how they looked closer into finding these motor proteins that help the chromosome kind of click, click, click along and start climbing across that microtubule. So that's how the chromosomes move. In metaphase, they line up in the middle. Pretty easy to remember. Let's go to the next phase. We have anaphase. Anaphase, the chromatids are going to start moving apart. They're moving along those spindle fibers with kinetochores, and we have an equal division. So pretty easy. The chromatids are going to split at the centromere, and they're going to start moving toward the poles using kinetochores. Finally, we get to telophase. Telophase, the chromosomes are now going to return to their thin form, so they're going to return back to chromatin, and you can see the nuclear membrane is going to start to reform, and we really have one cell that has 2 different nuclei. So we need another process to occur. This is the end of mitosis, but you have one cell with 2 nuclei. We don't want that. So what needs to happen is once the chromosomes return to that thin chromatin form, we're going to have a process known as cytokinesis occur. And that's where a cell just kind of pinches in, and then breaks apart. That's cytokinesis. And it forms this thing called a cleavage furrow. It's kind of like taking your hoodie and taking the strings, and if you pull the strings, the hood would collapse. That's kind of what the cell does. Those spindle fibers go and collect, and it causes the cleavage furrow to just pull in. It's kind of cinching closed, and you get those 2 new daughter cells. They are genetically identical. You've undergone tissue repair or growth. And that's what cytokinesis is. It's the pinching in of a membrane to form 2 new daughter cells. Now, you might be, thinking ahead and wondering, well, how does a plant cell do this? It has a cell wall. Cell walls are way too rigid. They're not going to be able to pinch in. So because plant cells have that cell wall, they can't undergo cytokinesis. Instead, they're going to grow a cell plate through the middle, and that cell plate will get longer and longer and longer until it connects with the outside cell wall, and you'll get those 2 individual plant cells. What I want to do is show you a quick little video that shows all of this unfolding, because they're not clear-cut stages. It's kind of like a movie. It all unfolds, and the stages kind of blend in from one to the other. So this is what we'll see, all the stages here. Okay, and you can see it then separate. We'll go through that one more time, a little slower. And you can see the centrosomes moving across, the chromosomes lining in the middle, anaphase, they're moving apart, telophase, we've got the 2 new nuclei, now we just need cytokinesis to happen. And you've got the 2 new daughter cells, and they are identical. So if you look behind me, you can see this is a pretty classic general biology lab where we look at an onion root tip. This, of course, is a plant cell. You might find some cells in here that are starting to grow that cell plate to separate the two, but I'm hoping that you can look at this and find certain stages that you clearly recognize. All right, so if you look up over here, you might be able to find one that is in metaphase, so that would be this one right here. You can see that those chromosomes are lined up in the middle. Here, can you recognize this phase? They're moving apart, so this would be anaphase. The rest, look at how many cells are in interphase. Remember, most of the time spent is going to be in interphase, so most of the cells should show signs where you don't see the chromatin in the thickened form chromatids. Okay, here you might see prophase over here. Okay, we've got those chromosomes shortening and fattening and ready for division. Okay, so that's it. That's mitosis. It creates 2 new daughter cells. They're identical. We have interphase, how it starts. It goes through a G1, which is a growth phase. Then it goes through S, which is the synthesis phase, where you replicate that DNA. And then it grows one more time at G2. And once all of those checkpoints have been met, it goes through mitosis, where we have prophase, metaphase, anaphase, telophase, and we get the 2 new daughter cells. So I hope that helped, and I'll see you next time.